analysis case. It's a bad one. Our goal is to determine a possible reason why COVID-19 is beginning to affect children, causing something called multisystem inflammatory syndrome, or MIS-C, which presents itself like the Kawasaki disease, but it also has other symptoms, some of which can be even more dangerous. After a spike in COVID-19 cases, there seems to be a correlating cluster of the MIS-C cases. I think to understand why it is happening, we first need to figure out how MIS-C affects the body. Exactly. The definition of MIS-C is an individual under 21 presenting with a fever, laboratory evidence of inflammation, and evidence of a clinically severe illness requiring hospitalization with multi-system organ involvement. Most children who have MIS-C test negative for a current infection with COVID-19, but many had a positive antibody test. What is the exact pathophysiology behind this? It may have to do with how our immune system distinguishes self from non-self. Normal cells express receptors called MHC that display peptides of all of the proteins expressed in that cell. T cells have a receptor, a TCR, that interacts with the peptide-presenting MHC molecules. If all peptides presented are self-peptides, the T cells just move on. If viral peptides are expressed, the TCR and MHC will fit together like a lock and key, activating the T cell, which will A, kill the virally infected cell, and B, expand the T cell carrying this TCR if there are more cells with the ability to kill the virus. It's possible that a super antigen that T cells are reacting to is cross-reactive with normal human proteins, causing the T cells to attack uninfected cells, which is causing the symptoms. Every T cell has a unique TCR that is generated during development. T cells containing TCRs that recognize the self-antigens are weeded out in a process called tolerance. During tolerance, T cells interact with thymus cells that express most of the genes in the human genome. Rather than expanding, T cells that recognize these peptides are eliminated from the T cell population. However, the development of autoimmune diseases, like lupus, tells us that tolerance is imperfect. To test the hypothesis that the superantigens induced by the coronavirus are causing this problem, one possible experiment would be to sequence the T cell receptors from the patients who are suffering from MIS-C and from patients who recovered from COVID-19 but did not present the symptoms of MIS-C. We would look for an expansion of a specific TCR in patients suffering from MIS-C that is not present in patients without MIS-C. Our investigation shows T cell receptor sequencing is a viable way to discover why MIS-C is affecting children.